Okay, so remember in the last video we talked about how mistakes happen during the DNA replication process, and how DNA polymerase actually has proofreading activities, this 5' to 3' exonuclease activity that can correct these mistakes. So in that video we learned that mistakes happen about 1 in every 10 to the 4th or 10 to the 6th nucleobase additions. Tautomeric forms or wobble pairs between nucleobases can lead to the addition of non-complementary nucleotides to the growing newly synthesized strand, and that these mistakes can be detected by DNA polymerase, passed into a 5' to 3' to 5' exonuclease site on the polymerase, and then removed by that exonuclease activity, giving the polymerase a second chance to correct the bases. And as I alluded to, we now know how DNA polymerase works. We know how it can proofread the DNA synthesis process to make it more accurate, but we still need to place it in the context of copying the entire genome. And it turns out that's going to be a little bit more complicated than polymerase activity itself, because a lot of things are need to happen in order to be able to copy both strands of our genome successfully. And so we're going to explore the helper activities that are needed to solve certain problems that arise during genome replication in this video. And so let's think about why replicating your genome requires more than just strand extension. Okay, so if we have a piece of double-stranded DNA and we're going to unwind it in order to be able to copy it, well, the fact that we need to unwind the double-stranded DNA is something that we need to think about, because this is going to require energy, as the double helix itself is a very nice stable structure. We just spent multiple lectures talking about how it's this amazingly super stable structure. And so we're going to need an enzyme that's going to be able to help us unwind this double-stranded DNA to give us access to the individual strands of the parental genome that we're going to use for synthesis. And remember that polymerase can't start off of on its own. It needs a primer to extend off of. And so once we've unwound these strands, we're going to need to put some primers down in the 5' to 3', 5' to 3' here, so that we can have something for the polymerase to extend off of. So something's going to need to put those down. And then if we think about this process, so if we are trying to say unwind in this direction and synthesize, copy both strands, well, if we keep unwinding, then in order to polymerize this bottom strand here in this region, we're going to need to add another primer, right? Because the 5' end is here. So we can't polymerize in this direction. Remember, polymerase only goes in the 5' to 3' direction. So in order to make more of this strand, we're going to have to actually put another primer down and synthesize off of that. And so now we're going to end up with multiple primers on one strand if we want to do this. And that's going to also create a problem, because what we're going to have is multiple fragments here. We're not going to have one beautiful complementary strand. We're going to have a bunch of tiny fragments. So we're going to need some enzyme that can join these strands together. And so it seems like at the bare minimum, to be able to copy both strands of genetic material like this, we're going to need a bunch of different enzymatic activities to be able to do all these little extra steps that we kind of deduced must occur in order for this process to work. So let's learn about those activities. And so the first type of activity we need to know about is something that's going to unwind the DNA. And you should come to understand that genome replication, the process of copying our genome, begins at what are called origins of replication. These are sometimes called ORIs by people in the who know the jargon. And these are locations where the DNA is going to get melted and unwound locally into 2 single-stranded pieces to provide access to the strands for synthesis. And the enzymes that are responsible for melting and unwinding DNA at these origins are called helicases. And what they produce is a resulting replication bubble, so this kind of stretch of single-stranded DNA on both sides that's going to provide the template for synthesis. So you'll want to know that helicases melt and unwind DNA to produce these bubbles. Now, once we have single-stranded DNA, we need something for polymerase to be able to extend off of. We need a primer, otherwise DNA polymerase can't extend. And so in order to do that, there's an enzyme called primase that's function is to synthesize a short complementary RNA sequence onto the single-stranded pieces here. And so you get this little DNA RNA hybrid that's made by primase, and this is going to be something that DNA polymerase can extend off of. And it's also worth noting that this hybrid is going to have a lot of A-form character to it, which is going to make it easier to detect and eliminate later on, because we're not going to want pieces of RNA in our genome. We're just using these little RNA primers here to get the process started. Okay, so now we've opened up the piece of DNA using helicases. We've got two single strands. We've got primase adding primers on it. And from here, we can start trying to replicate the DNA. And if you were to just picture this and kind of draw it out, within the replication bubble, you've got two directions you can go. You can go this way, or you can go this way. And in each case, because synthesis occurs in the 5' to 3' direction, you're going to end up with this phenomena that we kind of discussed a couple slides ago, in which one strand is going to be able to undergo continuous synthesis. So the strand moving in the same direction as the fork, once you've gotten a single primer on there, you can just extend and go all the way and keep growing as one big strand. However, the other strand, the bottom strand here, as the fork moves, continues to move on, every time we want to synthesize new nucleotides there, because synthesis can only occur in the 5' to 3' direction, we're going to have to put another primer down and extend off of it every time this fork moves. And so in this picture here, for example, you see one fragment where we had a primer and the extension of some strand in the 5' to 3' direction. But then we needed to put another primer down here and extend. And as this fork moves, we're going to have to generate another primer and extend, another primer extend, and another primer and extend. And so this bottom strand here is called the lagging strand, and it undergoes what is called discontinuous synthesis. That is, it's made up of its synthesis is made up of multiple fragments. And each one of these little fragments that's produced here is called an Okazaki fragment. It's called that because it's named for the guy that suggested their existence. And so in order to completely synthesize this lagging strand correctly, we're going to need a few additional activities. So if we think about the process so far, on the lagging strand, we've got to have primase come down and put a little 5' to 3' RNA primer like you see here. And then we're going to extend that primer, or rather we're going to use that primer to extend. And this extension process is performed by a particular flavor of DNA polymerase called Pol3. And it's going to extend until it encounters the 5' end of another Okazaki fragment. And once it hits there, it's basically going to fall off. And so primase and Pol3 working together are going to produce the Okazaki fragments like you see here. So in order to complete the synthesis of the lagging strand, we're going to need to do a couple more things. So as Pol3 and primase work together to make fragment after fragment after fragment, Pol1, another flavor of DNA polymerase, is a special polymerase that can actually recognize the RNA primers in the lagging strand, and it's going to remove them and replace them with bona fide DNA sequence. So basically, wherever there's this red coming along, Pol1 is going to come in here and replace that RNA with DNA. However, even when Pol1 has done its work, there's still going to be these gaps between the different DNA fragments. And so we have these what are called nicks, where there's discontinuities in the newly synthesized strain as it is annealed to the parent strain. And so we need another enzyme to seal these nicks together, and that enzyme is called DNA ligase. And so chemically what DNA ligase is doing is, if you were to draw, say, the starting material in the product, you've got one end here, you've got your nick where you've got 5' to 3', you've got a 3' hydroxyl here, and then at this end you have your 5' phosphate DNA. And so what we need to be able to do is connect the 3' OH to the 5' phosphate to generate sealed DNA like this. And the enzyme that's going to do this, as I mentioned before, is called DNA ligase. And it's going to turn out that this ligase is actually going to need the energy of ATP in order to be able to catalyze this reaction. And if we go through the mechanism of how DNA ligase works, it'll become very apparent why we need ATP for this to happen. So if we were to walk through the steps of the DNA ligase chemical reaction, the first thing that ligase does is it takes an ATP molecule, which, as you know, is a high energy nucleoside triphosphate, and it attaches a fragment of the nucleotide to itself. So this lysine residue on the enzyme, a base catalyzes nucleophilic attack on the ATP, and what forms is an enzyme AMP intermediate. So the enzyme actually has this monophosphate nucleotide monophosphate attached to this lysine here. So why would it do that? Well, the ligase AMP conjugate here is a good substrate for the phosphate of our nicked DNA to attack and kick off, or sort of, what should I say? The phosphate attacks the connection between the enzyme and this AMP, and now we get this AMP transferred onto our nicked DNA. So at this point, the ligase has been fully restored, and the AMP has been transferred to the 5' phosphate of the nicked strand. And of course, what this has done is it has created a very high energy good leaving group here to allow for the 3' hydroxyl of the other half of our nicked DNA to undergo base catalyzed nucleophilic attack of the phosphate, kicking off the AMP in the process and resulting in this sealed DNA. And so basically, now that we've charged the 5' phosphate with this good leaving group, the AMP, the reaction can go forward. And you should note, if you just look at this slide instead, you can't have the hydroxyl attack plain old phosphate here, because it would mean that like O- is a leaving group, right? It's not going to happen. You need to put something else on there to make a good leaving group. And I think you'll also see that in principle, this reaction is not so different from at least this step in the reaction here of the 3' hydroxyl attacking the phosphate. It's not really that different from the 3' hydroxyl attacking a DNTP. In both cases, what you need is a good leaving group on your alpha phosphate here in order to drive the reaction forward. Okay, so the bottom line from this discussion here is that genome replication needs a coordination of many different activities in order to allow the complete process of genome replication to occur. And so what I would recommend if you're trying to learn this material is to either be able to draw this kind of replication fork diagram like you see here, or at the very least be able to point at everything on this diagram here and explain what it is and why it's needed for the process, right? For example, why do we need DNA ligase on this bottom strand here? Why is that needed? What is it doing here? What is primase doing? So on and so forth. And in the companion video, I'll just show you how I like to internalize this process by drawing it out. I find that many of the activities, you can kind of deduce that they are needed. You can deduce that they're needed, and so then it helps you remember that you need a primase or you need a ligase and so on and so forth. Okay, so there's a few last activities that we need to just mention for completeness sake in order to facilitate the replication. One of those are topoisomerases, and so you can imagine, if you remember our demos we did with DNA topology, when you try to unwind DNA, it can result in the formation of these supercoiled structures. And those supercoiled structures, you can imagine if you're the replication machinery trying to move through them, these big superhelical structures could be disruptive to the process of replication. And so this is something where the cell uses topoisomerases to make sure that as these supercoils form during the replication process, it uses topoisomerase activity to undo and relieve that topological problems that are arising. The other useful activity to know about are proteins called single-stranded binding proteins. So you might imagine that if you pull those DNA strands apart, even if you used energy, you use a helicase to rip them apart, given enough time, they're going to want to come back together and reanneal, because it's favorable to have the strands reanneal to one another. And so to prevent single-stranded DNA from reannealing or annealing to itself, for example, there are proteins called single-stranded binding proteins that help maintain the single-stranded DNA that's produced from helicases in a single-stranded form. So they kind of coat it and protect it from reannealing. And so the last thing I'll mention about the replication process is that although we have discussed the leading and lagging strands kind of very distinct from one another in terms of all the challenges that come with synthesizing the lagging strand of DNA, I'll mention that the current model for how the replication fork moves and copies DNA is actually that both the leading and lagging strands are synthesized in a very coordinated fashion. And I don't expect you to be able to necessarily draw this, but the idea is that by placing the strands of DNA and the components of primase, helicase, your polymerases, and all those components in a geometry like this, it actually enables for you to conveniently synthesize both the lagging and leading strand in an almost completely coordinated and simultaneous manner. So that's just something interesting to note, is that it seems very awkward to us, but by adopting this geometry, it occurs very streamlined, actually. Okay, so there's one last thing that we should discuss. We've talked about everything that goes into the replication fork, the activities, the primases, the ligases, all of that stuff. But something else to think about is what happens when these replication forks get to the ends of chromosomes, and in particular lineochromosomes, the types of chromosomes that we eukaryotes have. Well, if you were to think about it, the fact that we need to remove those RNA primers always from the lagging strand is going to cause this problem at the very ends of that molecule, which is that we're going to have these gaps that we couldn't fill in because we've removed our primers and there's nothing to template off of. So something's going to happen if we don't do something about this. Then every time we copy a chromosome, the lagging strand is going to get shorter and shorter and shorter with each round of replication. And so the cell's going to have to do something about this, or eventually you're not going to have any chromosome left. And so it is the enzyme called telomerase that's going to provide a solution to this shrinking ends problem. And so what is telomerase? Well, telomerase is responsible for the construction of telomeres. And what are telomeres? Telomeres are regions of repetitive sequences that are placed at the end of eukaryotic chromosomes. So you can kind of think of these as sequences that cap the ends of our chromosomes. And telomerase is the enzyme that's responsible for putting on these telomeric sequence. And the way that telomerase gets around that primase problem that we talked about is that it actually carries a fragment of RNA along with it. So you can see here in this schematic, telomerase has this little RNA that it's bringing around with it everywhere it goes. And that RNA fragment is going to basically provide the template to perform DNA synthesis on the ends of the parental strand of DNA here. And once you tack on a little bit more of the sequence, what you've done is you've extended this far enough that now the standard replication machinery can come back in and fill in the original gap that you were worried about in the first place. And so this combination of telomerase bringing its own template and extending, and then the standard replication machinery being able to go in and fill that in, together is what prevents chromosomes from shortening after each cell division. Okay, so to just sum up DNA replication, and if you're curious about what that coordinated leading and lagging strand synthesis looks like. This is video over here playing. But the key things you need to know are that replication is going to begin at these origin sequences, and it's going to result in a replication bubble in which you've got replication forks moving in opposite directions. The unwinding and synthesis of DNA at these replication forks creates problems that helper enzymes are going to be needed to resolve. So those are things like primases and ligases and so on and so forth. You'll want to know that at these forks, there's a leading strand of DNA synthesis, which undergoes continuous synthesis. Is very easy to understand. And there's a second strand, the lagging strand, which is more complicated to understand. And it's made as a series of discontinuous Okazaki fragments that need to be sealed together by DNA ligase. And finally, you should know that when we get to the end of the chromosomes, that lagging strand once again presents more problems. And in particular, absent some solution, the lagging strand is going to shorten with each replication cycle. And so there's this enzyme telomerase, which is going to extend the telomeric sequences that cap the ends of the chromosomes and provide a way of preventing the shortening at the end of the lagging strand. Okay, so just to sum up everything that we've talked about in this lecture so far, this lecture series number 3, the key takeaways that you really want to make sure you understand are the chemical mechanisms for DNA synthesis. You want to know about that 3' hydroxyl attacking that nucleoside triphosphate, and that means that synthesis is going to go in the 5', 3' direction. You're going to want to know how DNA polymerase works and the requirements for it to be able to synthesize. And so the key things to know are it requires a primer, that is a short single-stranded DNA fragment annealed to target DNA to extend off of. The synthesis requires a 3' hydroxyl so that you can add the next DNTP. You're never going to add a nucleotide to the 5' end. That's really important. You should know that polymerase also contains a proofreading activity that can correct for mistakes. And in the context of genome replication, these constraints on DNA polymerase activity are going to lead to a lagging strand and a leading strand in the replication fork. And then finally, you're going to want to know about the different activities, the helper activities that are needed to overcome the challenges of actually copying the complete genome. So you're going to want to know about helicases that unwind. You're going to want to know about the topoisomerases that relieve supercoiling. You're going to want to know about DNA ligase that's going to connect those Okazaki fragments together in the lagging strand. And you're going to want to know about telomerase, which prevents the ends of linear chromosomes from shrinking with each round of replication. And so hopefully, through the 3 videos that we've got, you're going to be able to know everything on this slide very well, and it'll be straightforward for you to learn all this material.